Welcome back to part 6 on our series of DNA and RNA. Today we are looking at DNA mutations, which is any change in the DNA code. So, if we were to have DNA such as C, T, C, G, any change to these letters or even the sequence of these letters, if you add something, get rid of something, switch a letter out for another, take two of these and swap them. Any change in the order of the letters and what letters exist would be a change in the DNA code, and that would be considered a mutation. So there are 3 main ways that we're going to look at mutations. The first type of mutation we're going to look at is a substitution. So, for our example, we are going to use the same DNA in all 3 of our examples. The first one, our base DNA is ATC, CTT, GCA. So we have substituted something. A substitution is taking one letter and switching it with another letter. So in this example, we have taken the second T after the C and switched it for another C. So instead of ATC, CTT, GCA, we now have ATC, CTC, GCA. We have switched one letter for another letter. Our second type of mutation is called an insertion. And that's where you take a letter and you just put it into the code and move everything down a little bit. So, for this example, we have, again, ATC, CTT, GCA. Now we have ATC, A, CTT, GCA. We have taken a letter and we have put it right here in front of the C. So we have not changed any of the original letters. Those letters are all the same, but we have added an extra one within the code. And finally, deletion, which is the opposite of an insertion. If insertion is inserting a letter in, deletion is taking a letter out and not replacing it. So, again, ATC, CTT, GCA, and that leads us to ATC, CT, GCA. That second T, we have just completely taken out. This is where it would normally be, but we have taken it out. It does not exist. There is no letter there to replace it. So the 3 different types are substitution, changing one letter for another, insertion, putting an extra letter in the code, and deletion, taking a letter out and not replacing it with anything. And these, we're just focusing on single letters. These can occur at any level. You could have a substitution where 45 letters in a row get replaced with something, or 45 letters get inserted here, or an entire section of DNA, hundreds or thousands of letters get deleted. We're just focusing on what they look like at the single letter level. And why that matters is, why mutations matter is what we looked at during translation in part 5. Translation is turning that mRNA code into the amino acids, and eventually those amino acids combine to make proteins. Mutations affect the amino acids during translation. So, for example, here's our new DNA code. ATA, CTT, GCA. We then, through transcription, make our RNA molecule. UAU, GAA, CGU. And when we translate that, we get the amino acids tyrosine, glutamic acid, and arginine. Now, how the different mutations will affect stuff. First, substitution. We take CTT and turn it into CTG. We have switched one letter with another letter. That changes the RNA then, from GAA to GAC. And when we look at the amino acids, GAC does not code for glutamic acid. GAC codes for aspartic acid, a completely different amino acid. Sometimes you will get lucky and the letter change will result in the same amino acid. In that case, it is called a silent mutation. You don't see a change in the amino acids. But in this case, we did have a change. Now, let's say that instead of a substitution, we have an insertion. So let's go back to base. Instead of a substitution, we have an insertion. So CTT, GAA, glutamic acid. Instead of CTT, we are going to insert I need to write that in green so you know what we have changed. CTAT. We have added an extra letter. Now, the big difference that makes is before we had us already separated into our codons, our three-letter words. We have now changed the codons. And that change results in CTA being our new codon, and then TGC. So when we go down here and translate it into the RNA, instead of GAA, we get GAU. And then we would push there we go. Then we would push that other A over here, because now that T is in this codon and the A matches there. That then will change these two to, once again, aspartic acid and threonine. So we have now changed two codons, which is in itself a big change. Now let's go back, and unfortunately this time something very drastic has happened. And we now have a deletion. We'll put a dotted 0 where that deletion happens. So now where we draw is we go C, T, but there's no letter here, so we now must draw our codon here. And that codon, this color, once again, I don't know why I'm dotting that, gives us aspartic acid. It just happens that all of our changes result in aspartic acid here. So that is how the 3 different mutations can affect the DNA code and the amino acid sequence. So hopefully this has been much more enlightening if you're having trouble with any part of DNA, RNA, be it the structure, or how you copy it, or how you make RNA, or how you read RNA to make proteins, or what mutations, any change in the DNA code makes. Hopefully these videos will help you understand them. If you need help understanding how we are reading the RNA to get the amino acids, and how to read the chart that I've given you, the next video and the final video in the series will focus on how to read that RNA chart.